Student Invited Speakers Seminar. It's a great honor to introduce Simon Brandl, our Student Invited Speaker. Just a brief background on Simon. He did his PhD at James Cook University. He worked on functional niche partitioning and with David Bellwood, and then he did a marine geo postdoc at Smithsonian. And now he's advancing postdoctoral fellow at Simon Brandl University. So,
thank you, Samuel. Yeah, it's a real pleasure to be here, and I'm truly honored to have been selected. Thank you, everybody, for getting me to talk here today. I gather this is, the third seminar in not too long a time frame that is going to be about coral reefs, but I suspect it's going to shake out fairly differently from the previous two speakers. So specifically what I want to talk about today is I want to talk about fishes, functions, and the future of coral reefs. But before I do that, I want to talk a little bit about the riveting topic of personal finance. Now, obviously, I suspect nobody in the room here is an accountant or in finance, and nor am I, but it's fairly easy. There are usually two types of accounts that we have our money in. One of them could be a checking account that sort of has the day-to-day turnover that we engage in. Our salary goes in there, and we use it for grocery shopping and everything. And then we have a savings account that sort of is more the longer-term accumulation. And the idea here is that everything that is left over from our checking account eventually goes into our savings. And then over time, those savings build up and will make for a nice piece of real estate somewhere. But, of course, if you're a millennial like me, your savings account might not be all that opulent because you spend way too much money on avocado toast. So the bottom line here is that I really like avocado. However, sage financial advice would be for me to monitor my checking account to make sure that my savings account actually increases. So that would be the rationale for every reasonable financial consultant. Now, we're all biologists, so let's be done with the financial stuff. And as biologists, we might be interested in sort of the general state of an ecosystem, like this beautiful forest landscape in New Caledonia. And what we do to kind of assess sort of the overall health or resilience or state of an ecosystem is we're monitoring some variables. And those variables might, for example, if we're interested in rainforests, we might monitor forest cover, we might monitor the abundance of sharks on coral reefs, we might monitor the biomass of elephants in the savanna, or we might monitor the extent of penguin colonies in Antarctica. And that's fantastic. And the sort of general narrative is that sharks are really great, and the more shark we have on coral reefs, the better. But we all know that in order to get a lot of shark, you need to produce that shark through a very non-delicate process of predation. And what this means is that, in effect, when we're looking at tree cover or sharks or elephants or penguins, we're looking at the piggy bank. We're looking at this savings account of nature where lots and lots of biomass is stored away fairly permanently. In a sense, these are the savings, and this is underpinned by a much more dynamic checking account of nature, which are these ecological processes such as predation, herbivory, or nutrient cycling. This is the dynamic checking that underpins the savings on a day-to-day basis through the turnover. Now, there are some issues with this, because as I alluded to, most of our monitoring and conservation efforts are focused on this savings account, on the iconic species that we can see and observe in nature. Now, the reasons for that are fairly simple. For starters, it's much easier to monitor the extent of forest cover than it is quantifying primary productivity. The second reason is that soliciting funding and getting people interested in conservation is a lot easier for penguins than it is getting people to be excited about penguin poop. And so with all of this in mind, we have to realize that perhaps it's time for us to focus on the checking a little more and not just on those big glossy variables that are in fact the savings account. Problem being, of course, that beyond the monitoring and the funding issues, it's actually fairly intricate to determine these ecological processes, and they require knowledge across ecological hierarchies. For starters, and I'm going to illustrate this using an example of coral reef herbivory, which I've worked on during my PhD. For starters, we need to know the different species. So we want to know what a certain species feeds on, what resources it uses, or its ecological niche. We then want to know, hey, how do the different species assemble into communities? What are the coexistence patterns? What animals, what species are we likely to find in a certain place? And then finally, we can take this and say, hey, how does that affect an ecological process such as herbivory? And that is ultimately going to tell us what underpins the savings account in a certain place at a certain time. Now, coral reefs have the benefit that we've got a really long history of study, and we really know a lot about coral reefs compared to other systems. But they're also nice and clear, and they're warm, and it's a lot of fun doing fieldwork there. But unless you've lived under a rock and didn't attend the past 2 seminars by coral reef ecologists, you would also know that coral reefs are a little bit on struggle street right now because of global bleaching events and these kinds of things. And the most persistent call in the face of all of this is that coral reefs need to be managed for functionality. And ecosystem functioning really is what is this checking account that I've been talking about. And surely on coral reefs we have this figured out. The answer is we don't. Even though we have a lot of increase in sort of the literature on biodiversity and ecosystem functioning on coral reefs, we don't actually know what a functional coral reef looks like. And if we look at how these 3 disciplines, sort of the species, the communities, and the ecological processes integrate in this literature base, we can see that, hey, there's a fair few in each of these sections. But the overlap, where people actually try to build the performance of ecological processes on coral reefs from the bottom up, are far and few between. And what's even worse is that perhaps we don't even know what kind of processes we're looking for on coral reefs when we're saying, hey, this is coral reef ecosystem functioning. So what I've done recently with some co-authors is to actually define these processes on coral reefs, to say these are the processes that underpin ecosystem functioning on coral reefs. And they actually turned out to fall into these 4 nice reciprocal pairs of production and consumption of anorganic or organic material. We have bioerosion and calcification. We have herbivory and primary productivity. We have secondary production and predation, and nutrient uptake and nutrient release. And I would argue that these 4 are in fact the cornerstones of reef functioning. And for the rest of the talk, I will be focusing on this section right here. And that also implies that I will be focusing on coral reef fishes. Coral reef fishes, of course, are one of the best known aspects of coral reefs, and everybody loves these large mobile fishes. But today, my talk is actually going to focus on all of the fishes that you can see in these photographs. Now you're going to say, actually, except for that little grunt in the back there, I'm not going to talk about that one. Now you're going to say, well, Simon's lost it completely already. That's not quite the case. I would actually argue that these photographs are full of fishes. We just can't see them. And the reason for that is that they're about that big. This is what I would call a cryptobenthic reef fish. And so cryptobenthic fishes, as you can see, come in all shapes and colors, but they only really come in 3 size classes, which is small, smaller, and smallest. So from here, this is just a rough outline of what I'm going to be talking about for the rest of this seminar. I will first give you an introduction about cryptobenthic reef fishes, what they are, what they do, and so on and so forth. And then I'll highlight 3 sort of separate case studies on each of these ecological disciplines, the niches, the communities, and the ecosystems, all with their corresponding colors. And then I'll risk a bit of an outlook of what that means for coral reefs and cryptobenthics. So let's talk about cryptobenthic reef fishes. Now, what are cryptobenthic reef fishes? For starters, and I have to say this at this institution, cryptobenthic reef fishes are, for example, this one, which is the only vertebrate genus that is named after a university, the triple fin UCLA Xenogrammus. However, of course, for a definition, a group of fishes that is kind of like the one that is named after a university is not all that great a definition. So let's talk a bit more about what cryptobenthic fishes actually are. The first definition was by a guy called P.J. Miller in 1979. He was the first one to use that term, and he said they're small-bodied fish that exploit restricted habitats where food and shelter are obtained in or in relation to conditions of substrate complexity and or restricted living space with a physical barrier likely to be interposed between the small fish and sympatric predators. Right, so that's a bunch of gibberish for essentially saying these are small fishes that like to hide. And people have taken that definition and tried to develop it a little further. Dybczynski-Bellwood in 2003 said, hey, a cryptobenthic fish has to be smaller than 50 millimeters body size. And that adequately describes an anemone fish. And then Kovacic et al. In 2012 said, well, in order to be a cryptobenthic fish, they have to live within the reef. So they have to be sort of in the framework, and that adequately describes a moray eel. Now, if you fail to see the similarities between moray eels and anemone fishes, you're not alone. So do I. And so what I did recently is to actually try and come up with a quantitative definition of what cryptobenthic fishes actually are. And what I did is I went into the size distributions of 58 coral reef fish families with 6,288 species within them. And I sort of quantified the size distribution within each family. And then I set a wonderfully arbitrary threshold and said, if a family has more than 10% species that are smaller than this 50 millimeter size threshold that Dybczynski and Bellwood came up with, then that shall be considered a cryptobenthic fish family. And it turns out there are a few of them. And if you look at this graph, there are a few really nice things that we can see. First of all, there are 17 families in blue right here that are in fact cryptobenthic fishes that have more than 10% tiny species. Up to, for example, the triple fins up here, which have almost 90% of species that are smaller than 50 millimeters. So these are, as a matter of fact, the Peter Pans of coral reef fishes. They're the ones that never want to grow up. They permanently live in this size class that is usually reserved for juvenile fishes. The other great thing that we can see here, arbitrary as that threshold may be, it actually produces some really nice separation among these families. And we can see there's this big old tail that doesn't have any families at all with species smaller, that doesn't have any species smaller than 50 millimeters at all. And then we have a few that are sort of on the cusp and somewhat questionable. And I'm not going to argue that there are no cryptobenthic fishes in these families. However, just to get a sort of bird's eye definition, I'm going to argue that these 17 families are the core group of cryptobenthic fish families. Now we might be interested in, hey, how many cryptobenthic fishes are there? And to really get a handle on that, we have to look at description rates over time. So these are the cumulative description rates of large coral reef fishes since the 1750s, sort of divided into these 4 periods over time. And we can see, yeah, there are a few bumps along the way, but generally it's been a pretty steady climb of description rates. Now, if we put cryptobenthic fishes on there, it looks a lot different. They were extremely slow to get out of the gates, and then kind of once we hit the scuba diving and the DNA time periods, they're absolutely skyrocketing, essentially showing an exponential increase. And we can do a little bit of modeling with this as well, where we can say, hey, what's it going to be like in the next few years? And we can essentially calculate the ratio between cryptobenthics and large reef fishes, and we can find out that by 2031, if the description rates continue as they have been over time, there will be more described cryptobenthic reef fishes, around 4,000, than large reef fishes. So there is a ton of diversity in this group of fishes. Now, the reason they were so slow to get described is because finding them in an environment that looks like this is like finding a needle in a haystack, of course. So how do we, in fact, sample those fishes? What we're doing to get quantitative estimates of cryptobenthic fish communities is by using what we call closed anesthetic stations. So what we use is a big bell-shaped net that is weighted with chain in the bottom. We put that over a little reef outcrop and then follow this with an impermeable tent. This is just literally the top of a tent with the bottom cut out. And then we spray a solution called clove oil, which is an anesthetic for fishes, into that area. we lift up the tarp, and then we gently fold back the net and collect all of the tiny little fish with forceps and come away with a nice little bag full of cryptobenthic fishes. We can then have a look where that kind of quantitative census has actually been employed in the world. And what we can see, these are the blue dots right here when we're talking about the tropics. It's lacking both sampling depth and sampling breadth, with some areas like, for example, the Indian Ocean, just a small ocean anyways, not being sampled at all. When we look at the numbers, these are densities per square meter. We can find that wherever people have looked for cryptobenthic fishes, they have shown up in droves. This, for example, in the Red Sea is an estimate of 30 individual fishes per square meter. So that's about the size of the tops of one of these chairs, 30 individual vertebrate animals on that area. So it sounds like there's a lot of cryptobenthic fishes, and it sounds like if you are a cryptobenthic fishes, you're A, beautiful, and B, you also have lots of friends. However, life out there is, of course, fairly hard. And so the main challenge for us all and for organisms in general is you want to find enough to eat, you want to avoid getting eaten, and you want to reproduce. So these are the main three challenges. And what we'll see in a second is that for cryptobenthic fishes, these are non-trivial at all. In fact, there are extremely hard challenges for these animals. What I'll show you is three graphs that are all on the log-log scale with body mass on the x-axis and respective variables on the y-axis. And in the dots are families, in blue we have cryptobenthics, and in white we have large reef fishes. The first one is mass-specific metabolic rate. So what that means is, and we can see that cryptobenthics generally are farther up than the rest of the reef fish families. And what that means is that per gram body mass, cryptobenthic fishes actually have to work harder to feed themselves. That doesn't mean that they need more food. As a matter of fact, a whale shark will still consume vastly bigger quantities than cryptobenthics, but for each gram body mass, they have to work harder to maintain just their basic regular functions. What that means is that they have to spend an awful lot of time foraging. This is a Fusigobius that is devouring a worm. And this is a herd of Gnatholepis caurensis in a tank, and you can kind of see what they do. This is what they can do all day, just sit there and munch on the sand to meet their high mass-specific metabolic rates. The second one has mortality rate on the y-axis. And this is the get eaten part. And what we can see is that cryptobenthics are spectacularly bad at not getting eaten, meaning they die all the time. So this is a triple fin that is about 2 inches or an inch and a half, eating a small dwarf goby that is about the size of your thumbnail. So even within cryptobenthics, there's an awful lot of predation going on. And some of these cryptobenthics, they're all annual species, so hardly any cryptobenthics will live longer than a year, and some of them will have an adult lifespan of about 3 weeks. And then we have reproduction. And we have the cryptobenthics down here in this case, and that means that their clutch sizes, we have clutch size on the y-axis, are extremely small. That means they only put out a few eggs for each batch of eggs they produce, especially compared to things like luganids or ceranids, which produce thousands and thousands of eggs. Whereas some of the smallest cryptobenthics, like some of the ghost gobies, for example, this is a yellow sponge goby on a yellow sponge with yellow eggs, will produce as few as 34 eggs per batch. So that's quite remarkable. So, with all of this, it seems like life as a cryptobenthic isn't necessarily a picnic in the park. And that means that they will have to have evolved some fairly unique life history strategies to cope with all of this. And so far, I've kind of talked about cryptobenthics as a whole group that kind of does the same thing, all of them, which is very unlikely to happen. And that leads me to the first kind of case study, which is the study of ecological niches in cryptobenthic fishes. And to talk about that, I will take you to Caribou Key in Belize, which is a small island on the Belizean barrier reef, which is exactly as big as it looks like on this photograph. This is Belize, and this is what the reef looks like here. And in green, we have those kind of exposed forereef sites, and then in the pink, we have the sheltered back reef sites. And these are two fairly distinct habitats on the Belizean barrier reef. And I focused on 4 species in this study, which are 2 triple fins, Enianectes altivalis and Enianectes matador, and 2 tube blennies, Acanthemblemaria spinosa and Acanthemblemaria aspera. The 2 triple fins are generally known to be these sort of benthic foragers, so they'll scoot around the benthos and pick on things, whereas the tube blennies are known to forage mostly on drifting plankton, while they stay hidden in their tube with just their head sticking out. Now, if we look at sort of the general literature and just look at the 2 families, the triple fins and the tube blennies, we can see that really both of them eat a little bit of everything. These are dietary items garnered from the literature on the left, and these are different species in these respective groups, and we can see very, very little differentiation among them. However, of course, if you've ever looked at the gut contents of a fish, you will have been less than impressed, because it's usually a little bit of half-digested mush. And the only reason that can be made worse is if that half-digested mush is about 2 milligrams. So if you can think of these invertebrates, they turn into a completely indistinguishable matter within a very short time period, and that will exacerbate us finding out what these species actually feed on. So what we can use is DNA metabarcoding to actually get a precise handle on what these fishes eat. In a sense, all you do is you take out the gut contents of a fish, you extract all the DNA that's in the guts, and then you sequence that, and you essentially match the resulting operational taxonomic units, or OTUs, to these potential prey groups, and you get a really detailed picture of a species diet. And what we can see here, this is just a network based on sort of the higher order taxonomic affiliations of the prey, and we can already see a fair bit more differentiation between the 4 species than we saw previously. So for example, we have Enianectes altivalis, which seems to be the only one that feeds heavily on polychaetes. We have, for example, Enianectes matador, which seems to like the neogastropods quite a bit. So there's a fair bit more differentiation already. But keep in mind that these are just broad taxonomic prey groups right now. We actually have OTU-based data on this. So we can create this network tree where the 4 colored nodes are each of the 4 species, and then we can just look at, for example, crustaceans as prey items, and each symbol here is a separate OTU. And what we can see is that there's really strong differentiation between the 4 species, and all 4 of them seem to have their unique clouds of prey items that they seem to feed on. And the other thing we might see here as well is that some of these clouds are a little bigger than others. So we can also look at the diversity of prey items that these fishes ingest. And that's where this study got really interesting. Because for both species pairs, the triple fins and the tube blennies, we had one species that had a higher OTU count than the other on average. And so we have Enianectes matador and Acanthemblemaria spinosa, which I'm going to call the high diversity feeders, or generalists if you want to employ that term. And then we have the respective partners, which feed on average on fewer OTUs, which are Enianectes altivalis and Enianectes aspera, or the low diversity feeders, or specialists if you wish. And then we can look at the distribution of these species across these sites on the Belizean Barrier Reef, and that's where it gets really interesting. If we take the exposed sites, which were the ones in green, we can see that there's no difference between the high diversity feeders right here and the low diversity feeders right here. But if we go to the sheltered sites, we can see that the high diversity feeders drop out completely, as in they just don't live there. And so that gives us a really interesting hint that these fine differences between species dietary niches seem to be driving these distribution patterns of the species on the Belizean Barrier Reef. So to recap that, we have these really sort of subtle differences that we have to employ really advanced techniques to get at them, that show that there is a fair bit of partitioning in these species. And then we find that these trophic differences seem to also relate to the distribution patterns of the 4 species. So that would suggest that, hey, perhaps cryptobenthic fish communities are mediated by the environment and the niches of the respective species, and we might see some strong patterns in the community assembly of cryptobenthics as well. And that leads us to communities. And to talk about that, I'm going to lead you to the United Arab Emirates, which have lots of shiny buildings and some less shiny coral reefs. But we also have some cryptobenthics on these reefs. And specifically, I'm going to take you to two regions, both in the United Arab Emirates, in the Arabian Gulf, here in blue, and in Fujairah, which is on the Gulf of Oman side, in pink. Now, those two regions differ quite massively, even though they're just about a 3-hour drive through the desert apart. The Gulf of Oman is a perfectly reasonable coral reef environment, with summer maximum temperatures of about 32.6 degrees centigrade and winter minimum 23.0 degrees centigrade. Whereas the Arabian Gulf is actually the most extreme coral reef environment we know in the world, with summer maximum temperatures of 35.2 degrees centigrade and winter minimums of 16 degrees. So naturally, there might be a fair bit of difference here, and what's really interesting about this part is that this is a really interesting kind of laboratory for what the future might look like. With climate change and everything, the temperature is supposed to rise, so hey, if we look at this, can this give us a glimpse into the future of coral reef systems? So we did a bunch of fieldwork, did a bunch of collections on both of these sites. And after lots of collecting and photographing, we come up with these nice, beautiful cryptobenthic fish communities. And we can look at the community composition. And perhaps not surprisingly to anybody, they are fundamentally different. We have the Arabian Gulf communities right here and the Fujairah communities right here. And as you can see, they're fairly distinct. Now, 3 species actually occurred on both sides. So they were present and abundant in the Arabian Gulf and in Fujairah. But then in Fujairah, we also had a bunch of other species, and I'm just highlighting 3 for the purpose of this, that were present in Fujairah, but not in the Arabian Gulf. And the reason for that is, of course, easily derived. One would expect that those that are present on both sides have a higher temperature tolerance than the ones that are not present in the Arabian Gulf. And we can, of course, test this by using something called the critical thermal maximum and minimum. So essentially what you do is you put a fish in an aquarium, and then you ramp up or you turn down the heat until that fish goes belly up, and that is the critical thermal maximum. It's not a very subtle technique, but it usually does a really good job defining a species' physiological temperature tolerances. Now, if we look at that, for these 6 species that I've highlighted earlier, we can first see that, hey, the species that occur on both sides, yeah, they seem to have a fair bit of difference in their thermal tolerance, but we can also see that the Arabian Gulf side in blue, those populations seem to have a higher temperature tolerance than the populations from the Gulf of Oman. And we can also plot what is sort of a collective temperature tolerance in those populations. And then we can look at the other species, and this is somewhat puzzling, because while there's still a fair bit of variation in them, at least 2 of them seem to be fairly capable of withstanding the same temperatures that those species that occur in the Arabian Gulf can tolerate. As a matter of fact, if we put in the collective thermal tolerance, it is no different whatsoever from the collective thermal tolerance of the species in the Arabian Gulf. What's more is that the actual maximum summer temperature would be right here on this graph. So what I take from this is that critical thermal maximum, which is our gold standard for testing a species' tolerance to heat and what is used in most of our climate change predictions, is not useful at all when it comes to predicting whether the species is going to be there or not. So essentially, temperature doesn't have any effect on the composition whatsoever. And nor does size of a fish, the trophic group, or the phylogeny, which is data I don't have on this presentation. Instead, I would wager that it is much finer scale variables such as diet, microhabitat affiliation, or metabolism or behavior that are going to drive these differences in composition. And what we'll see in a second is that these differences in composition also have an effect on other community metrics such as diversity, abundance, and biomass. As a matter of fact, the Gulf of Oman, which is the standard coral reef environment, has about twice as many species, about 6 times the density of individuals, and about twice the biomass of the reefs in the Arabian Gulf. So that could be a really interesting sort of harbinger of what's to come in the future. And the interesting part here is seeing how these communities affect the larger scale, higher order variables, such as biomass. And that leads us to actual ecosystem scale processes. Now, you might have sat through all of this and said, yeah, you know, Simon, I really don't do small fish. I'm much more interested in large fish. Why do I have to care about cryptobenthics? And my answer to that is, if you do like large fish, you better start caring about cryptobenthics as well. And the reason for that is found when we look at the larval stages of cryptobenthic and large reef fishes. So what I did is I went through the literature and I essentially did a meta-analysis of the larval composition near coral reefs worldwide. And what I found is that in every major biogeographic coral reef region in the world, about 60 to 75% of the larvae that are around in the plankton are cryptobenthics. So that means that they are a huge proportion of this larval pool that is available to coral reefs worldwide. Now you might say, well, Simon, that's just because they're so abundant. And as a matter of fact, yeah, you might think that larval supply, or what is around in the nearshore ichthyoplankton, might very well scale with the gamete output of the adult populations. And you know, we might have large reef fishes down here with lower output and cryptobenthics with really high output up there. However, if we actually use data rather than this completely hypothetical model, we see that, yeah, there is a fairly weak relationship between adult gamete output and larval supply in large reef fish families. So, you know, for everything they put out, they get a little less back, but there is a positive relationship. Cryptobenthics, however, play a fundamentally different game. And as a matter of fact, if you calculate the regression coefficients for that interaction effect between for cryptobenthics, it's about an order of magnitude higher. It's about 2 orders of magnitude higher than for large reef fishes. And what that means is that for every egg that these cryptobenthic fishes lay, they get much more bang for their buck than the large reef fishes. And that has some really interesting ecosystem scale consequences that I'll walk you through. And this is a theoretical population model that we ran using these data. And it all starts with this larval supply. This is the sort of 75% contribution of cryptobenthics to the larval pool. We can go from there and say, hey, these larvae are going to settle on the reef. And at settlement, we find that, okay, the large reef fishes are a little bigger and might account for a little more biomass, but generally not much difference in the settlement stage. Now we've settled those larvae on a coral reef. And now we can let them grow and we can let them die using empirically derived data. And when we do that, we find that there's fundamentally different mortality and fundamentally different growth between the two groups. And so we can look at the net produced biomass. And the beauty here is that cryptobenthics don't account for anything. They're, you know, maybe 1/6 of the net produced biomass compared to large reef fishes, even though they have many more larvae settle on the reef. But the true twist of events comes when we look at the consumed biomass. So this is your day-to-day turnover that is quantified as the consumed fish biomass on coral reefs. And we find that cryptobenthics account for more than 60% of that consumed biomass. Or in other words, any large fish on the reef will be about 60% cryptobenthics at that point in time. And then, of course, if we spin this further, we can look at standing stock biomass, which is the single most commonly quantified metric of ecosystem functioning and secondary production on coral reefs. And we can see that cryptobenthics, again, don't account for anything. So in other words, these cryptobenthics are the checking account. These are the fishes that produce a ton of the consumed biomass, but because they're so small and because they die quicker than we can actually census them, we don't see them in the most commonly monitored metrics. In other words, this is the cryptocurrency of coral reefs. Pun totally intended. Now, the question is, how do they do that? What's driving all of this? And the answer for this probably lies in the dispersal. So if we think about this, how do they manage to have these prodigious numbers in the nearshore ichthyoplankton? We might arrive at the conclusion that, hey, they just don't go anywhere. And this kind of dispersal of marine organisms is the hallmark of our understanding of marine populations. And my suggestion is that cryptobenthics, as a whole, there might well be species that do this, but cryptobenthics, on average, don't disperse. They will hatch from their eggs, they'll swim off the reef, they'll drop to the bottom, and that's where they'll stay. Whereas the vast majority of other larvae will go out into the pelagic environment, which is a nasty, nasty place to be. They will die, and only few of them will eventually return to the reefs. And that's, in my opinion, the strategy that cryptobenthics employ, and that's what makes them such a critical functional group on coral reefs. If that is the case, then we should see some patterns that, if we think about that in a macroevolutionary context, we should see the following pattern. We should see extremely high species richness, because very little dispersal leads to extreme population differentiation, and over evolutionary time to lots of speciation. However, we should also see that that strategy is fairly rare, because it is prone to extinction. If you don't have the lifeline of pelagically dispersing larvae, your populations might very well just go extinct in the blink of an eye because the conditions change. And as a matter of fact, if we look at cryptobenthic fishes on the fish tree of life, we see that in blue right here we have two major origins of being cryptobenthics, which is the Blenniformes and relatives on the left here, and the Gobiaria, which is the gobies and apogonids right here. But both of those clades are extremely diverse. As a matter of fact, they're two of the most rapidly diversifying clades of bony fishes in the world. And so that matches this hypothesis of larval dispersal. And that leads me into the final part, which is the future. And there's something about this that is really quite concerning. Because if you think about this, that means that most of the cryptobenthic species we know might not have this pelagic lifeline, and that they, for many reasons, might simply go extinct if there's a slight change in conditions, and we have a local extinction, and we don't know where they're going to come from to repopulate that area. And that gets worse if we think about how we monitor and how we conserve animals like cryptobenthics that are small, that are part of this checking account, and that are not the iconic, large savings account species that we usually tend to focus on. Now, this is data comparing the size distributions of all fishes in freshwater and marine systems to the size distributions of monitoring events in the single biggest time series of marine of fish monitoring, which is the Biotime database. And we can see that the Biotime database focuses on greatly bigger species than there are actually in nature. So there's clearly a bias towards larger fishes in our monitoring efforts. And we can also look at the probability of being data deficient in the IUCN database. These are just fishes again, and we can see that there is a much higher probability of being data deficient if you are smaller than there is if you are a bigger fish, which again makes sense. The other thing here too is the way we assess vulnerability. Vulnerability of species is most commonly assessed in terms of the vulnerability to exploitation, especially in fishes. So in fishes, we might look at generation times and sort of the growth and the output, the reproductive output. But all of this is fairly irrelevant if your main threat is not overexploitation, but things like habitat change and coral bleaching. So again, our sort of assessment criteria aren't tailored to small animals. They're tailored to large, iconic animals that are suffering from human exploitation. And if we then think even further and think about the conservation efforts that we employ, and in the marine systems that is primarily marine protected areas, these are all optimized to benefit things like sharks and parrotfish and butterfly fish. They're not optimized to benefit cryptobenthic fishes. Now all of that is very easily explained because we generally have a stronger attachment to these kinds of species than we have to cryptobenthic fishes. And this is a matter of being rational. This is a matter of being pragmatic. And even us as scientists perhaps are about as pragmatic as I am about buying avocados at the store, which cost $2.50 in Vancouver, by the way. So perhaps the best way of putting it is by Baba Dioum, a Senegalese ecologist and conservationist, who says, in the end, we will conserve only what we love. However, there is actually a second part to this quote, which is, we will love only what we understand, and we will understand only what we're taught. And I hope that I was able to make the case today that it's our responsibility as scientists to actually focus and hone in on these underrepresented groups that don't get monitored, but that for what we know might be absolutely critical to ecosystems such as coral reefs. And with that, I'd like to thank everybody for your attention, and if there are any questions, please feel free. Probably turn on the light too if you want to.
Questions?
Yeah. Wait. Go ahead.
So it's kind of got me thinking of, I was trying to think of like a terrestrial analogy to cryptobenthic fish. And I'm curious if you thought about that. Are there other small species that we don't pay attention to that end up being a huge productivity?
Yeah, I mean, the first place you would go there obviously is insects, which we know have a fundamental impact on terrestrial ecosystems that are the bottom of many trophic pathways in the terrestrial realm, and that we know have suffered from a fair bit of disturbance over the past decades, starting with insecticides in the 1950s, 1960s. There was some recent controversy about the extent of the insectocalypse or something, the actual rate at which insects are going extinct. But that would be one place to go. They don't really fit any of the other categories, and they don't fit the insects all that well either. I guess rodents might be something reasonably similar. They are vertebrates. They are very small. They often have fundamental implications for the rest of the food chain. But this actual combination of being absolutely tiny. They're the smallest marine vertebrates we know. The only reason they're not the smallest vertebrates is because there are a couple of species of frog that are shorter than cryptobenthic fishes. If you go by mass, cryptobenthic fishes are by far the smallest vertebrates there are. With them being so small and being so abundant and having such high turnover, these extremely short lifespans, that, as far as I'm aware, doesn't have any parallels in the vertebrate world. Sure.
Do you see any seasonal variation in diets when you look at them? So in other words, are there food facts and food mean times that might help explain kind of diversity?
That's really hard to tell for starters, because a lot of them won't live through the seasons. So the same individual, you won't ever be able to observe any differences over time.
But even within, say,
within a species, I suspect there are some differences. So what we see with a lot of the metabarcoding data is that a lot of fishes will be extremely opportunistic. So if there's, for example, if the damselfish are spawning, you can bet that the triple fins will have damselfish DNA in their guts because they're chomping away on the eggs. So there's certainly that opportunism that is kind of famous on coral reefs, that these fishes are so versatile. But even through that, I mean, these fishes were collected over a 3-week period, and we've just published a paper today in Methods in Ecology and Evolution that had a bigger data set of larger fishes. And even with all that kind of seasonal variation and the temporal stochasticity of spawning events and mortality events and plankton blooms and whatnot, you still get these fairly distinct niches for each species. So I think that the staples will always be something that the species is fairly unique in, and then you supplement that with whatever is available at the time. Yeah.
Do adult cryptobenthics have reduced or absent cast bladders?
Yes, so a lot of them will. Most gobies, for example, don't have a swim bladder at all. I don't know if they have a swim bladder as larvae, and I would suspect they probably don't. If anything, they probably have some oil droplets or something that would give them some buoyancy, if anything. There are a couple of species that live on the underside of caves, sort of upside down their entire lives, that will have a swim bladder, but it is probably actually secondarily evolved in these species, because the vast majority of gobies do not have swim bladders.
That would be a parallelism across these diverse groups then?
The lack of swim bladders?
Yeah.
Yeah, so how well do you think that the sort of the macro fish community of coral reefs that we do think about in conservation context and we do protect the MPAs and stuff, how well do they act as umbrella species for the small benthic guys?
Right.
And, because that's a
Sure.
common strategy.
Yeah.
Right? And oftentimes, especially given the food web that
Yeah.
you described, you might well predict that places that are very diverse for larger fishes will also be very
Right. So that's a really interesting question that I'm thinking a lot about. So I'm certain there are some parallels that makes marine protected areas beneficial, period. But I think the key here is that when we think about optimizing these, there's a lot of conflict that arises from the interactions with humans in the implementation of marine protected areas as it pertains to large reef fishes. So we want to make them as big as possible because large reef fishes have larger home ranges, and we want to make sure we protect them. I think if we were to focus on small cryptobenthics, we could implement, one square kilometer protected areas that actually do a decent job maintaining and conserving this high productivity environment. And as a matter of fact, if you go to a place like Fiji, for example, there are a lot of these community-based closures, which are literally, they might be half a mile across. And the difference is absolutely breathtaking between those two. And so I think, while I'm not arguing against using large fish species as sort of the umbrella species in this context, and I think there is a need for that because they are the ones that are heavily exploited. I think we should diversify in how we think we can conserve coral reefs and the actual dynamics of coral reefs rather than just the species. And for example, I think habitat complexity is probably one of the biggest drivers for cryptobenthic fish abundance and diversity from what I can tell from my data. And so thinking about how we can preserve complexity or how we can recreate complexity once it has been lost is a very useful exercise that we can engage in to keep that kind of that heartbeat of coral reef fish productivity going. Yeah.
I really enjoyed your talk. Thank you. My question, I had a couple, but the first one was when you presented the results about the clutch size of eggs, and I forget what the other variable was, but I was curious if you accounted for parental care in those comparisons, because a lot of those cryptobenthic fishes are nest brooders.
Yeah.
They have parental care, whereas the large reef fishes are broadcast spawners.
Mm-hmm. That is correct. So I didn't in that particular log regression. That was just part of a review to show the general patterns. In the actual adult gamete output versus larval supply analysis, we actually did account for being benthic spawners versus broadcast spawners, and that it's a massive difference, of course, because they're fundamentally different strategies. But there's some crossover in each of these groups, as in damselfishes, for example, are benthic brooders. Some of the tetrodontids will be benthic brooders, and vice versa. There are some cryptobenthics that are actually broadcast spawners, such as callionymids. And it didn't improve the model whatsoever, putting that in as a factor because of these crossovers. So yes, different strategy altogether, and certainly an important factor in driving the number of eggs that are being spawned, but not necessarily a great explanatory variable for what we actually did.
I have one more quick question. It's,
Sure.
Okay. Okay, sorry.
All right. Sure. Yeah.
Speaking to this idea that low dispersal might make them more vulnerable to extinction and probably higher endemism. Has anyone looked at population structure or anything like that, and maybe I'm sort of thinking we would expect lower, maybe effective population size versus their larger than counterparts, and is there any evidence of that?
Yep. So yes, there have been a few studies. For example, one done on the Australian Great Barrier Reef, Gerlach et al. 2007. They did a study of population structure in 2 species of cardinal fish and 2 species of damselfish. They're about even in size. Maybe the cardinal fish is a little smaller, but the 2 cardinal fish species had much higher population structure across, I think, a scale of probably 4 or 5 miles. Than the 2 damselfish, and but sort of in the same vein, there's a study on the median population, median dispersal of Elacatinus gobies. These are cleaning gobies in the Caribbean. Daloya et al. In 2015 in PNAS, and they showed that the median dispersal kernel of Elacatinus lori, I think, is about 1.3 kilometers. So that really speaks to this kind of difference in dispersal. And as I said earlier, not all of them do it. For all we know, there are some species, whether they are actually one species or not, I'm not sure, but there are species that are distributed from the Red Sea to French Polynesia. But as far as I'm aware, nobody's really looked at the structure of these, and I guess exceptions make the rule. So, yeah. Yeah.
Does that dispersal limitation ever factor into regeneration efforts? I mean, for the replacement of artificial substrates, for instance, wouldn't that need to be close enough so that cryptomentics would be able to colonize?
So this is a really interesting question, and something that I'm hoping to get at in the future, which is what drives these populations? Does it literally just take a couple to at some point completely stochastically arrive on a reef and then just build this booming population? Is that enough to bring them back, or is it not? So that would determine whether you can enhance cryptobenthic fish populations through the provision of, for example, highly complex structure on a coral reef. What I see in my research is that if I go resample a place such as Lizard Island in Australia, we went there 2018 and did a bunch of collections all around the island, and we had data from 2011, 2012. Yeah, from 2011 and 2012. And I think the 5 most common species in 2011, 2012, none of them were there in 2018. And the 5 most common in 2018, none of them were there in 2011, 2012. So there's an extreme kind of boom and bust dynamic going on. And I think in a system like the Great Barrier Reef, where the next reef might only be 3 or 4 kilometers away, that's fine. But it does worry me for places like oceanic islands, where nothing is around for hundreds and hundreds of miles, and then what? So yeah, that is certainly something I'm interested in, and that hopefully in the future I'll get to.
But on that, sorry, do you have? Are there data that from Micronesia that show that there have been extirpations or extinctions?
There are some data. There's one paper, Monday et al. 2004, that shows the local extinction of a coral goby species, which obviously that's one of the first ones to go, because they're specialized on a certain species of acroporic coral. But the problem, as I showed in that talk, is that we have almost no data whatsoever on on these fishes at all. And I suspect that if I were to, this is one of the projects I'd like to do at some point if I can find a parallel universe in which I have some time. If I were to go through all of the recent sort of descriptions of cryptobenthic fishes from certain places, and then document whether or not they are actually being picked up again in later monitoring efforts, I would almost be convinced that the data will show that they aren't, and that there is a lot of extinction going on that we are not currently documenting, just because they're so small, they have such restricted ranges, and nobody cares about them.
Sorry, I remember my question. That second experiment where you had the temperature test, my question is if you measured dissolved oxygen in the water, and because I know gobies, like, they can breathe air often, and if they observe them breathing air, and if that was a strategy.
Right. So I guess the question is, would air capacity to breathe air, would that potentially allow them to exist in the high temperature environment? Is that?
Yeah. So like if the water gets warm enough, that the dissolved oxygen goes down and the fish will die. But
yeah.
I know like gobies often can breathe air.
Right.
To survive in like no oxygen water. So
yeah.
I was just curious because it showed that some of the, like there was variability in the species that you use, but it seemed like from back here it looked like it was the goby that was surviving.
Right. Yeah. So we didn't measure dissolved oxygen in this particular one. There is evidence of these dead zones in the Arabian Gulf. So oxygen limitation may very well be an issue. I think you're right in that the Hetereleotris and the Coriogallops, which were the two goby species and that kind of had the highest tolerance, they are probably able to kind of shut themselves down a little bit and go into these kind of torpor-like states, and that might help them. But it still doesn't explain why the Hetereleotris, which is actually fairly close to the Coriogallops, does not occur in the Arabian Gulf. So by that reasoning, those two goby species should both be doing well in the Gulf, but they're not. One of them never occurs in the Gulf. So, yeah. Any other questions?
All right, well,
All right.
let's thank Simon again.
Thank you.